Hi there. This is part 3 of the genomics section of the course, and I'll be talking about DNA virus genomes today, which are on average larger than RNA viruses, and some are pathogenic and others, most are not. So here's the graph for DNA viruses, also RNA viruses below that, and in general, DNA viruses can reach a larger length than can RNA viruses, primarily because of the differences in mutation rates. So I'll be talking about several DNA viruses, starting with VX174, which is a single-stranded DNA viruses. All the others are double-stranded DNA viruses. So here's bacteriophage VX174, the life cycle, or the infection cycle. It consists of 5,368 nucleotides, single-stranded DNA virus, and it infects strains of E. coli. So starting up here on the left, the virus attaches to the surface of the bacterial cell through a receptor which is recognized by the H protein. And then it injects the single-stranded DNA minus strand into the cell. The A protein from the virus then is a polymerase, a DNA polymerase, makes a double-stranded molecule. And from there you get transcription of the RNAs. Those mRNAs are then translated on host ribosomes to make part of the virus particle into which the DNA will be threaded. In addition to the A protein, the A star protein is made, and in conjunction with the proteins C and J, causes rolling circle replication of the genome. So it threads out single-stranded copies of the genome, which are then threaded into the head of the virus, and then one of those proteins clips a genome's worth of the DNA off, and that gets fully encapsulated. And then the H protein, which is the protein that recognizes the cell surface of the bacterium, is installed on the outside of the virus particle. And then the protein K from gene K is the thing that controls the cell burst size when the cell is going to break open. And gene E forms a protein that lyses the cell. So although this DNA virus has a very small genome, it's nonetheless complex in how it infects and gets itself replicated within the host cell. As we work our way into larger genomes, think back to this VX174 and the small number of genes and the complexity with which it completes its infection cycle. As larger genomes will have similar systems, but just built one on top of the other until we get to cellular systems then that are much more complex, but have the same small beginnings. This is the VX174 genome. It's circular. When it enters the cell, it's actually packaged in the virion as a linear. There's the origin of replication, ORI, and then starting with the promoters, promoter A, promoter B, promoter D, and then you have terminators of transcription. And they're leaky terminators, so that you do get some transcription through those at various times. So starting with A, you can produce the RNA from gene A, but you can also produce an A star, which is the one that causes the rolling circle replication of the chromosomal DNA. You can get read-through C, D, and J, but then there's a terminator. But as I said, that's leaky, so that you can get some transcription through to F, and then there's another leaky terminator through to G, another terminator, and then finally through to H, where you get full termination. The other two promoters then lead to transcription of genes, of RNAs for genes B and K, and then another one for D and E, the D and E RNAs in proteins. So even though this is a small genome and a very simple virus, you can see that there's been fine-tuning through evolution to produce RNAs and proteins in the proper amounts at the proper times, so that this virus can go through its infection cycle, make new particles, burst the cell, and then those particles go on to infect new cells that have been uninfected. The second virus I'd like to talk about is T4, and it's a member of the T-even DNA viruses. So there's T2, T4, T6 viruses. There may be more than that. I know there's a T7 also, but that's a T-odd. So this has a head, an icosahedral head, a tail, and then tail fibers, both long and short tail fibers, as well as a base plate, all of which are used to infect E. coli cells. This is a transmission electron micrograph negatively stained preparation showing a cell that was unlucky enough to be infected by many T4 viruses. Note that the tails are shortened, and that's because when it injects the DNA into the host cell, those tails shorten just like a little hypodermic needle. The genome of bacteriophage T4 and all T-even viruses is quite a bit larger than FIX174, and this is a double-stranded DNA virus. In this case, it's about 169,000 base pairs. In addition to carrying the genes for tail fibers, tails, heads, and things to make the virus, it also carries with it genes for DNA replication, transcription, translation, some on nucleotide synthesis and DNA repair, lysis, some of unknown function, and then a couple of genes for inhibitors of the host restriction system. The restriction system meaning the restriction enzymes that would chop up foreign DNA. In this case, it has inhibitors of those systems so that its DNA does not get chopped up by the host enzymes. This diagram shows the assembly of bacteriophage T4, including some of the gene products, the proteins that are necessary for putting those portions together. So you can see the head, just to start the head, takes a series of 12 different proteins to assemble the head. And then two more genes, 16 and 17, are required to thread the DNA up into the head, and then host and sock then put a cap on that to hold the DNA inside. In the meantime, the tails are produced. The base plates, you can see, take 6 different genes, and then the wedges require another 7. The complexity of the base plate is primarily caused by the fact that these are what interact with the host cell membrane and cell wall, and so these are crucial to infection. And then other gene products put on the tail. The tail fibers are then also assembled, and then several gene products aid in putting the tail onto the head and then assembling the tail fibers onto the tail. So this takes a very coordinated set of reactions to replicate the chromosome of the virus, to transcribe the genes in the amounts that they're needed, and then in translation of those on host ribosomes to create the proteins and then assemble the virus. Incidentally, these viruses can be assembled in vitro also, just by making the parts, mixing them in certain proportions, and the virus will then self-assemble. So the next virus I wanted to cover is bacteriophage lambda. Again, it's a double-stranded DNA virus. In the virus particle, the genome is linear, which is on top. But once it's injected into the host cell, it circularizes. And so at the bottom there you can see where those COS sites, COS-L and COS-R, come together to make a circular molecule. And the COS just stands for cohesive end, so they have self-complementary ends that get together, and then the ligase in the host seals those up to form the circle. So on the left-hand side, you see head and tail genes for making the particle. On the right-hand side is a lysis gene for lysing the cell at the end. And then in the middle, there are all these genes primarily for control of expression of those genes. So this diagram just lays that out and sort of groups things so we can talk about them a little bit. The genome is 48.5 kilobases in length, containing 40 genes. And again, on the left-hand side, you see the head and tail genes, and then one gene for packaging. On the right-hand side, you see the gene for lysis. And then in the middle, you have a control region consisting of 9 genes, which also includes 4 promoters, 2 operators, 4 terminators, and 1 origin of replication. And then a couple of genes over here for integration, excision, and recombination. And then a middle section here, no one really knows what that section does, and when these are used as vectors, that part is cut out, and then recombinant molecules are made with different genes in that region. That is, that's where you would place genes of foreign origin that you want to place within the lambda molecule in order to make more copies of them. So once lambda infects a cell, it can do one of 2 things. It can either go through the lytic cycle, where it makes more copies of itself and lyses the cells, or it can integrate into the chromosome by lysogeny and create a prophage. That is, the virus is integrated into the host chromosome. So lysogeny is indicated here, where you first get circularization of the chromosome, and then there's a site on the lambda genome called @lambda, which matches up with an @B site on the host chromosome, and there's a crossover that occurs, and then the lambda is integrated into the host chromosome. So this diagram will show both pathways. This is the lysogenic pathway, where the linear chromosome is injected into the cell. You get the COS sites coming together. Ligase from the host seals that up into a circular molecule, and then you get the early genes off, integration genes on, crossover at the sites, and then integration into the chromosome, forming the prophage. At this point, the genes that would excise the chromosome and turn on the lytic cycle are inhibited from expressing. So at some point in time, usually if the cell is under stress, meaning nutritive stress or something else, or UV stress, DNA damage, then you'll get a switch to turning the early genes on, which would shift it to the lytic cycle. So it first excises from the host chromosome, circularizes, and then you get theta form replication, which is sort of the normal type of replication you see in bacteria. And then later it switches to rolling circle replication, which puts out a linear concatemer of the genome of lambda. And then the heads and tails are produced, and finally the DNA is threaded into the head, and then at the cos sites, these are cut by an enzyme, protein A, filling the head with exactly 1 genome's worth of the lambda chromosome. This shows the assembly and maturation of bacteriophage lambda, starting from the upper left to create the head. A scaffold pre-head is produced from several proteins from several genes, and then the GRO-E product finishes off the head, and the GRO-E is a host from a host gene. And then the proteins NU1 and A start threading the DNA up into the head, and they are flanked by the A protein actually sits on the cos sites, and once the head is filled with the DNA, it cuts at the cos site, so there's exactly 1 genome's worth of the lambda genome in each head. And then F1 changes the conformation of the head, and eventually you have the termination protein acting on that, just removing the rest of the DNA from the opening in the head. F2 plugging the end, and then the W protein then attaching the tail to the head. And in the meantime, the tail is being produced by several gene products, and again, it's fairly complex as far as the number of proteins that become part of that tail, and that's mainly because that's the very important part that will attach to the host during the infection stage. And then finally you have a complete lambda ready to infect a bacterium. As I indicated, that central section of the genome is a control region, and it's highlighted here. And things begin at the PM and PR region of the lambda genome. So this is a control region that if you go in the rightward direction there, you head towards the lytic cycle. And if you go off to the left of this diagram, you go into the lysogenic cycle. And so the proteins that are controlling that are C1 and CRO. And you can see that if you have CRO, it will inhibit leftward transcription. And if you make C1 will inhibit the transcription to the right at the PR promoter. Past that, you have CRO also affecting promoter PL, C1 affecting PL in a slightly different way. And then there are terminators, TR and TR1, TR2, etc. And those are affected by the N protein in a positive way. That means if N is produced, there will be read-through of those terminators, and so that you can get transcription past a normal termination site. So depending on the relative proportions early in the infection of C1 and CRO, that really determines the direction of transcription and whether you go towards the lytic type of growth, forming more lambda particles and lysing the cell, or whether you go to the lysogenic direction, remaining integrated in the host cell chromosome. So this diagram provides a little more detail on that process. And if you look in the center at that PRM-PR region, what you find is those two promoters overlap with one another, and they also have operator sites, OR1, OR2, and OR3. And C1 and CRO actually bind to those regions, sort of in a reverse order. So if you have CRO, it binds best to OR3 and a little less to OR2. And C1 forms a dimer and binds best to OR1, a little less to OR2. And so if you have C1, it will block the PR, the rightward promoter, and will inhibit the lytic pathway. But if you have more CRO than C1, then it will bind to OR3 and it will block the PRM promoter, and so it will inhibit the lysogenic pathway. And so you can see down below, if you start from C1 and CRO, you make some of each. If you get more, if you get read through to produce C2, that will lead towards the lytic pathway. However, if that pathway isn't as rapid and you make more of the N protein, that will stimulate transcription towards the left direction and the lysogenic pathway. The next virus I want to talk about is the virus, and it comes with a Sputnik satellite virus. Often there are many types of viruses that also have satellite viruses, in that the main virus can infect cells on its own, the host cells on its own, but the satellite viruses can only infect if the main virus is there. So this happens here with the virus and its satellite virus Sputnik, as well as with a virus called tobacco necrosis virus, which also has a satellite, and the satellite is pretty much a small version of the main TNV virus, but it's a plant virus. But it doesn't have some of the functions, so it can't produce virus particles on its own. However, when it is present with the main TNV virus, then it is produced in higher numbers than is the main virus. The viruses are very large viruses. In fact, when they were first recorded and observed, they thought that they were bacterial cells. And their genome size is also quite large, at 1.2 megabases, which is larger than many bacterial genomes. They also are unusual. They infect acanthamoeba, type of amoeba cell, and they set up their own vesicle. So they initially infect the cell, set up a vesicle, and then send the DNA into the nucleus. The DNA and the RNA, that is the transcribed RNA, is then sent out to these vesicles, which become virus factories. So they look just like organelles as they're being produced, but they do use the host ribosomes. So that's one thing. And then eventually lyse the cell and release all of the virus particles. When they infect with the Sputnik satellite virus, then they still set up the vesicle the same way that they would with just the virus infection. But the only thing that gets replicated are the Sputnik virus genomes. It still makes the same proteins, sets up the organelle, and starts producing virus particles, but only the Sputnik genome is encapsulated into those viruses, and then eventually it lyses the cell, producing all Sputnik virus particles rather than virus particles. So in this case, the Sputnik satellite virus is termed a virophage. That is, it's essentially infecting a virus. So it's a virus infecting a virus that's infecting an amoeba. And so it's kind of a complex system that's evolved in this case. These are transmission electron micrographs of infection by the virus. So you can see how it sets up that vesicle in the middle, and then you get the virus particles coming off of those. There's a virus compared to a mycoplasma, which is a bacterium, and you can see how large these viruses are. And they have this peculiar structure where they have these fibrils coming out from them also. So they're pretty unique in their size and morphology. Here's some electron micrographs also showing what happens when you get infection with the satellite virus or the Sputnik virophage. So a normal particle down here of the virus, and this one up here is when you get infection with the Sputnik in a virus infection. So you can see these small little circles here are the Sputnik virus being produced within a virus particle. Another interesting thing about this genome, in addition to its size, is that it has some genes that you don't expect viruses to have. So it has things for replication and transcription, which are common in viruses, but then it has genes for translation, cellular function, post-translational modifications, cell walls and cell membrane synthesis, nucleotide transportation, transport and metabolism, as well as some other genes that you would expect in a bacterium, in a cellular organism, but aren't expected in a virus. And so this has led to, some conclusions that these are just reduced bacteria, pathogenic bacteria, one more step in the evolution towards viruses or organelles or small particles of one sort or another. The Sputnik virus also has some unusual genes on it. A few are expected, like the major capsid protein and the integrase. Most of the other structures, the other genes, the positions aren't known, although it is known that there's a primase helicase that's one of the genes, an ATPase, a zinc ribbon protein, and an insertion sequence, such as what you would find in a transposon. Also, if you look between the genes, there appears to be quite a bit of secondary structure, mainly hairpins and loops. And so the structure is, some of it's expected of a virus and other parts are not expected of a virus. But it looks like this system might have evolved from a virus infecting a bacterium that infected an amoeba, and eventually there was reduction in those genomes so that now you have the virus, which was an ancient bacterium infecting an amoeba, and then it has this virus that is infecting the virus as well. So a pretty complex system, but it's interesting from an evolutionary standpoint. So that's the end of the discussion on DNA viruses. Next time I'll be discussing the initial parts of 2 presentations on bacterial genomes. See you later. Thank you.